Hello everyone, welcome to my channel, MCAT_crackers 007. This is Alex, a third year pre-med. My channel is tailored for all science students, high school and university, so please subscribe and stay tuned for science content. So today's topic is about enzymes, and the subtopics which I will cover are catalysts, enzymes as biological catalysts, types of enzymes and their mode of action, mechanism of enzyme activity, and regulation of enzyme activity. So first, what are catalysts? Catalysts are substances that speed up the rate of reaction without being used up in the reaction. And they favor endergonic and endothermic reactions where thermal energy is needed, like the input of thermal energy is needed for the reaction to happen. So with the help of catalysts, these reactions occur at room temperature or at reactions lower than required. And as we can see from this thermodynamic graph, with an uncatalyzed reaction, the activation energy is higher compared to when using catalysts. So the mode of action of a catalyst is lowering of activation energy to make the reaction possible to occur. And it increases the number, it increases the number of increases the number of successful collisions which are needed for reaction to occur. Next, we talk about enzymes. So enzymes are a type of catalysts which are made of proteins, hence why they are called as biological catalysts. So they have a primary, secondary, tertiary, and even quaternary structure, where the primary structure is made up of amino acids, and these amino acids form polypeptides, and these polypeptides form tertiary structures, these foldings we see over here. And when we have two or more tertiary structures together, they form a quaternary structure. All right? So moving on to types of enzymes, we have 6 types of enzymes. We have oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. First, in oxidoreductases, redox reactions are catalyzed. What are redox reactions? Redox reactions are reactions in which oxidation and reduction occur simultaneously, like at the same time. So reduction reactions are where oxygen is lost and electrons and hydrogen are gained. Oxidation is where oxygen is gained or is used, and electrons and hydrogen are lost. Example of oxidoreductases we have is a lactate dehydrogenase enzyme. Transferases, where when we have two reactants reacting together, so transferase enzymes transfer function group from one reactant to the other. Function group like carbonyl, function groups are like carbonyl, a thiol group, etc. Next, we have hydrolases. Hydrolases and lyases, they have a similar function in that they cleave a bond. Hydrolases cleaves bonds with the aid of a water molecule, while lyases does that without the use of a water molecule. Then we have isomerases, which produce isomers of the molecules. It produces constitution isomers and stereoisomers. Constitution isomers are isomers that have same molecular formula but different structural formula, and stereoisomers have different spatial arrangement, different arrangement in space. The last type of enzyme which we have is ligases. Ligase enzyme forms bonds between molecules. So ligases are used in anabolic reactions, whereas hydrolases and lyases are most likely seen in a catabolic reaction. Next, we move on to the mechanism by which enzymes work. We have two potential mechanisms. First, lock and key than induced fit model. Enzymes and catalysts in general work by the principle of the active site, which is a location on the catalyst, which is, we can say, which is the functional component of the catalyst, where the substrate binds, and it's the site at which the reaction is catalyzed. In lock and key, the active site is that site of the enzyme is complementary to the shape of the substrate, and hence the substrate binds to the enzyme at its active site as the key binds to the lock, thereby it's called lock and key mechanism. While in induced fit model, initially we have the active site of the enzyme non-complementary to the substrate, but when the substrate binds to the active site of the enzyme undergoes a conformational change, which results in the enzyme changing its active site and binding firmly and rigidly onto the substrate, as we see over here. So which of these two mechanisms has more effective binding of enzyme to substrate? Well, it's the induced fit model. And both of these mechanisms result in an enzyme-substrate complex where the substrate is bound to the enzyme. Enzyme-substrate complex is an intermediate product in the catalysis reaction. Next, we move on to how enzymes are regulated. So enzymes are regulated by two ways, one of which is called feedback inhibition. So feedback inhibition occurs by the following way, where we have a series of enzymes which are interconnected together by specific intermediates. So we have over here enzyme 1 and its substrate, which result in an intermediate, the purple one, which is the substrate for the next enzyme in sequence, which is enzyme 2. And then this is the substrate for enzyme 3. Now we have an end product over here, end product of the whole chain reaction. And this end product, when it's produced in excess amount, it accumulates and goes and binds over here to the enzyme at its allosteric site, a site other than the active site. This results in the enzyme, which is enzyme 1 over here, undergoing a conformational change that changes the active site in such a way that the active site is no longer complementary to the substrate, so it can no longer bind to the substrate. Hence, it's in a state of inhibition. The second mode of regulation is reversible inhibition. We have three modes of reversible inhibition. We have competitive, non-competitive, and mixed. So competitive inhibition is where the inhibitor binds at the enzyme's active site, preventing the binding of the substrate. In other words, it competes with the substrate for the enzyme active site. In non-competitive inhibition, we have the inhibitor binding to a site other than the active site, which is the allosteric site. And when it binds to the allosteric site, it results in the enzyme undergoing conformational change, that changes the active site and prevents the binding of the substrate. It's similar to feedback inhibition, but the difference is over here we are using an inhibitor, and in feedback inhibition, the inhibitor is natural. It's the end product of the reaction. Whereas here, inhibitor could be external drugs taken, or cofactors, metal ions, etc. And then to the third type of reversible inhibition, which is mixed. This happens when the inhibitor has unequal affinities to the enzyme and enzyme substrate complexes. And in that case, we have mixed inhibition. So this concludes our lecture for today. I hope you liked the lecture and how it's presented. This is my first video on YouTube and my first the first video in my MCAT_crackers 007 channel. Please like and subscribe and stay tuned for more content. Thank you and have a good day.